morning, everyone. It is such a pleasure to have you all here this morning. We are about to get started. My name is Brittania Moodie, and I am one of the leads here at Caribbean Youth Development Institute, and I am super excited to have you all here. We are going to hope that this is a very interactive session for you all. So if you have questions, please ask. If something isn't clear, please let us know. We want to make it as enjoyable for you as it is for us to have this moment to share with you guys. So I am gonna get started by introducing Ian. And Ian is my PI, but my mentor, and he's sitting right behind me. So he's he's our principal investigator. And what that means is he is in charge of everything that takes place or happens in lab. And he runs the show, writing the grants and ensuring that we have everything all set that we need. So Ian is going to introduce himself, and then I am going to introduce the next scientist when he appears. All right.
Good morning, everybody. Hi, my name is Ian. And as Brittania said, I get to run one of the labs here at Whitehead and MIT. And I can tell you a little bit about our science, but I think that the most important thing is that I get to work with Brittania, which definitely makes it an awesome place to be. Whitehead is a place where there's a lot of science that happens, and we're hoping to introduce you in a little bit to one of the other PIs here as well, Toby Ono. And actually, he's right there now. I just saw him join. Yes. And so actually, our game plan for this morning was that Toby can go first, because we're going to have a special treat today where Brittania and I are actually going to show you around our lab at Whitehead and show you some cells on the microscope, which is going to be a lot of fun. But before we do that, I think maybe Brittania talking to Toby a little bit would be great too.
Hey, Toby.
Hi, how are you?
We're good. All right. Thank you for joining us this morning and for giving us the time of your day. So I have here some very interesting students in, what happens in a lab? What does a scientist really do? We can see that we have around 36% of us who are at the primary level, and then 64% of us who are at the high school level. So that's great. That's amazing. And so Toby, we have here some very interested students, from K through 12, who wants to know a bit more about your research and kind of what you do. So I'm going to start by asking you to just introduce yourself and then tell us a bit more about your research program. What do you hope to achieve long-term by the work that you do? And then what are some of the day-to-day activities that actually take place in your lab?
Well, thank you so much, Brittania. This is great. I'm so excited to meet you all and to get to talk to you and share from you. My name is Toby. My full name is actually Tobaloba Oni, but that's a mouthful. So, so just call me Toby. So I'm originally from Nigeria, and I came to the United States about 12 years ago.
Okay.
It's been a long time now. And I really didn't know that I was going to be a scientist. I knew that I loved, I loved biology, in high school. I loved understanding what was going on in like a single cell. Because our bodies are made of all these cells.
Yeah.
And they do different things, and it just blows my mind that they can all work together. And look at me, I'm made up of all these cells, and they're all working together, and they work, and that always blew my mind.
Yeah.
But I thought I was going to do medicine. At least that's what my parents sent me to the United States to do.
Yeah, I think I can relate to that. And many students here probably can, right? Back home, a lot of us are, okay, we're either going to become, a doctor or a lawyer, maybe an engineer, right?
Yeah.
So I definitely can agree with that one.
Yeah, exactly. And so, yeah, I mean, I came to the States, started to study, and I learned more and more about the magic that happens in a cell.
Yeah.
And how, when it goes wrong, it could cause really bad problems.
Mhm.
You know, it could cause cancer, for example.
Yeah.
Cancer is basically when the cells in the whole body, you know, the cells just refuse to work together, and you know, a particular cell kind of does its own thing and goes crazy.
Yeah.
And so it made me really interested in understanding what that means and understanding how we can correct or fix that cell or get rid of it, so that you know, cancers that really afflict so many people in the world can be obliterated. So this is what, you know, kind of drove me to the science side of things. And you know, when I told my parents I wasn't going to do medicine, you know, they cried a little bit. But you know, now they support me, and they understand the importance of what I do.
Yeah.
And so to you know, tell you briefly about what I do, and I want to hear from all of you, and I want to, you know, field questions from you guys. But before that, I'll just quickly tell you, you know, about what I do. So I'm a fellow now at the CYD Institute, same as Ian, and I have the pleasure of like being right below Ian. And so I get some of that magical, you know, discovery kind of aura. But really, you know, so I started here a couple months ago, and my lab is really focused on something that you know, I find one of the most important things when we start to think of how can we really get rid of those crazy cells in our bodies, those cancer cells. And it's not easy because these cells are crazy, and they know how to, like, you know, make all the normal cells do their work for them. And so, you know, we've known for a long time that, you know, the immune system really helps us fight infections like COVID and you know, flu, colds, and all that kind of stuff, bacteria. But something we now really appreciate is the fact that the immune system has the ability to not only recognize, but completely eliminate those crazy cancer cells in our bodies. And so my lab is really focused on understanding how can we get immune cells to wake up and like fight cancer. And we're using
what are some examples of immune cells in the body?
so the you know, the soldiers in terms of fighting cancers are T cells, what you call T cells, and they're extremely powerful, and they have the ability to kill multiple cancer cells, you know, in basically one round. And then they replicate to high levels, you know, they grow, and then they form this mass of warriors that are able to actually really attack the cancer cells. And we understand now that there are 4 things that you need for immune cells, especially T cells, to eliminate the cancers. 4 things. And I say, you know, I call this, you know, the 4 things you need for immune cells to rage against cancer. One is R, which you need to recognize the cancer. So the T cells need to be able to tell between what is cancer and what is not cancer. A stands for activate. So the immune cells need to be switched on, particularly T cells. They can be active all the time, so otherwise they kill all your normal cells.
Okay.
So they need to be activated. And sometimes in many cancers, they're just sleeping, they're exhausted, they're not activated. So they need to be activated. The third thing is the G, which is they need to go get into the tumor. Because the immune cells, they're soldiers. They're not just sitting there in the tissues waiting. They're going everywhere, kind of like, you know, going through the lymph nodes and like just traveling around. But when they get activated, they need to be able to enter the tumor. And one of the things that we know now is that, you know, tumor cells sometimes form this thick nest around them, like a forest, and prevent the T cells from entering into the tumor. And the last thing is obviously eliminate. So the E stands for eliminate, because obviously you need to, when you actually get into the tumor and get near the tumor, you need to be able to eliminate the tumor cells. And tumor cells have found some ways to actually block the elimination. And so what we're doing in my lab is to go through those 4 principles of R.A.G.E and develop tools to activate all of those 4 things.
Awesome.
Increase the recognition, increase the activation, increase getting into the tumor, and increase elimination of cancer cells.
Awesome. That, I think for me, I think about these cells kind of being bullies, I suppose, telling the other cells kind of what to do and how to do it. And they have different ways of ensuring that work gets done, right? You spoke about the forest and all the things that it may create in order to actually stop our bodies from functioning properly. I have someone here asked a question, when people do transplant, I'm assuming like a blood transplant, can blood cells go into those little tubes? So I suppose the tubes that, you connect to your body, can blood cells actually go into those little tubes?
Yeah, so blood cells are very, they're shaped in a way that it's easy for them to like enter really tiny little capillaries. And so, they're particularly red blood cells, right? They're able to enter into this, into various, channels. But immune cells typically need to be activated in a way where they're able to actually attach to certain networks of capillaries or being able to squeeze in between endothelial cells so that they can actually enter tissues. And one more thing about transplant is that actually transplant is one of the things that in some cancers is very helpful, where you can take a patient that you can basically get rid of their you get rid of their blood cells, their bone marrow, and then you transplant the bone marrow of someone else, and that basically can activate those immune cells to kill the tumor in the transplant patient.
Awesome. All right, anyone has any other questions for Toby before we move on to the next question? Or did any of that make sense or not make sense? Or you want us to re-explain something or break something down for you? Okay, I see no. I see people nodding their heads no. Okay, all right. So you spoke about coming, from Nigeria to the United States, and kind of, briefly about your journey of getting to where you are right now. What were some of the things, academically and outside the classroom that you did that got you to this point? Because you said, initially you thought you were going to just be a doctor. So you're coming, I suppose, for med school and, ensuring that we have all those things. But what were some of the things that changed from a journey perspective that led you to here today?
Yeah, so, when I was in undergrad, I did research. And even though I wasn't doing, research on cancer at the time, and the research was actually quite simple. There were just like green algae. And we would put them under red light and they would swell. And we would measure, to what extent did the red light make them swell. So simple things like that were the things that sparked my curiosity even more about cells and about what makes them work and what, didn't make them work. So I think, getting early research opportunities, the ability to go to a small lab or go to a place where you're able to actually, feed on curiosity and actually be able to do some science in the early days were very important to me.
Awesome. And I can agree with that. And, that's why a lot of the things we do here at Caribbean Youth Development Institute is just try to get the kids involved from a young age and from a hands-on perspective, because I know a lot of students tend to learn better by using their hands, or they find it a bit more engaging than sitting in a classroom and just listening to what the teacher is saying, right? And so that's great. Anyone has any questions for Toby before we skip over to showing you guys a bit more about what the lab looks like? And we're gonna walk down the halls to the Cheeseman lab so that you guys can get a sense of that. Anybody has any questions at all about anything?
I thought actually that real quick, I mentioned earlier about, the forest that cancer cells sometimes make. And I have a picture here that I think will, do that a little bit of justice. And so if you don't mind, I'm going to share that real quick.
Yeah.
Okay, share screen, right? Can you see it?
Yes, I am able to see it. Are you guys able to see it?
Can everyone see it? Good.
Yes. Okay. I heard it.
So the blue you see here is the forest that pancreatic cancer makes, and it makes it really hard for immune cells to get in. The white arrows I'm showing you is actually the cancer cells. So you have this tumor where the cancer cells make up 10 to 30% of the whole tumor. The rest is this forest. And one way we like to think about it is, we think about this in terms of, oatmeal raisin cookie, where, the cancer cells are the raisins, and the rest of the forest is the oatmeal. And it really makes it hard for that G principle of getting into the tumor for the immune cell. So we're working on this to try to loosen the forest and change things around so that immune cells can get it. And on that note, I'll stop the share.
I see someone here has a question. Can cancer affect eyesight? Is that something that's possible?
Yeah, there are tumors that can affect eyesight for sure. But those tumors tend to be localized, and they tend to be brain tumors that, either kind of grow into areas where you perceive sight, or tumors that occur near the eye.
Okay. And someone asked, what are the stages of cancer?
Oh, that's a brilliant question too. So cancer tends to start slowly, and it actually happens that cancer develops over a long period of time. So the first stage is that, it grows within its own area. It starts to, what we call proliferate. It starts growing, and then the next stage is it starts to invade into other tissues, nearby tissues. And then when it invades into nearby tissues, then it gets into the bloodstream, and then it spreads around the body. So this actually correlates to the different stages of cancer in the clinic. So stage 1, stage 2, stage 3, and stage 4. Stage 1 being that early stage where it's still, growing within where it started from. Stage 2 being, well, it's bigger now, and it's starting to invade. And then stage 3, meaning that it's really invaded, nearby tissues, and it's entered into the lymph nodes. And then stage 4 being that it's in other organs, it's spread to other parts of the body.
Okay. And someone says, how do you decide if it's benign, but could be malignant?
Oh, that's another great question. Typically, benign tumors don't invade. They don't invade into nearby tissues. And so that's one of the key differences between benign tumors and something that is malignant. There are other ways we can tell based on what we call molecular characteristics of the tumor. But one of the key differences is that if it's benign, it grows slowly over time and just stays there. And then you can really cut it out. there are three major ways that we try to address tumors in the clinic. One way is to cut, so surgery. You can burn, so radiation, or you can poison, that's chemotherapy.
Okay.
And traditionally, you don't poison things that have not spread yet. If it's benign and it's staying there, you can just cut it, or you can also use radiation to burn it. But when you start to poison, typically that happens when the cancer is spread other places.
Someone says, does cancer usually grow, or where does cancer usually grow? Is it that it grows in the organ or healthy tissues? Is that area usually healthy, or what's the sense about where cancer actually localizes and stays? And how does cancer enter the body? Someone asked that question.
Oh, another great question. What makes cancer different from other diseases that tend to, infections, for example, and what makes cancer really hard to treat is that cancer typically does not come from outside the body. Cancer are our cells that go crazy. So because they're so similar to our cells, it's hard to find things that are different in cancer cells that we can treat. Typically, if viruses or bacteria infect us, they're so different that you can find something that kills the bacteria but doesn't kill us. But with cancer, it's harder because, finding something that kills cancer, which is our own cells that go crazy, it's really hard. And this is why the immune system is really perfect for finding out what is cancer, what is not. So the number one principle, the R, recognize. You have to be able to find out what is cancer, what is not. In terms of where cancer grows in the body, cancer can grow from most tissues in the body. One of the most common tissues that cancer grows from tend to be epithelial tissue, which are cells that tend to coat and surround and basically form the walls of a lot of our organs. So they line a lot of our organs, and they're typically, in interface with the environment. So they're getting damaged a lot. And those tissues tend to be more susceptible to cancer. And when cancer grows, it basically grows from being in a normal tissue, and then it can spread and, enter other normal tissues. So it doesn't matter if there's if you have surrounding normal tissues or not, cancer can grow in that environment and even spread to other parts of the body.
And someone said, how many persons, is there a statistic? How many persons can cancer kill a day? And what's the most dangerous type? And where does it affect?
That's a brilliant question. So it turns out that in the world, the number one killer is cardiovascular disease. So heart disease kills most people in the world. The second is cancer. And so it's a big, big burden. There are different kinds of cancer. So cancer is not just one single thing. There are, cancers that arise in the stomach, and the pancreas, and the lung, and the skin, and they're all different. And so the most common cancers are typically lung cancer, because people smoke a lot and all that. Prostate is very common. Breast cancer is common. But the ones that kill the most lethal cancers, pancreatic cancer, which I study, is actually the most common killer of people, especially in the United States, and also in the world, actually. And about 53,000 people die from pancreas cancer. Now, it's not as common as lung cancer and breast and prostate cancer, but it's super aggressive. And most people that have pancreas cancer die from pancreas cancer. Brain cancer is another one, particularly glioblastoma, very lethal, very lethal. And then you have some rare ones that are super lethal, like cholangiocarcinoma that arise from the bile duct.
Someone says, is skin or brain tumor, which is more dangerous, skin or brain tumors?
That's another terrific question. So this is a great question because skin cancer, particularly melanoma, used to be very dangerous, very lethal. But then immunotherapies came along, and these are therapies that wake up the immune system to kill the cancer cells. And melanoma, for example, now is not as dangerous as it used to be. Those therapies, unfortunately, don't work yet for brain cancer. So brain cancer is more dangerous currently. It's very dangerous, particularly glioblastoma.
If one more question, if cancer enters like the organs, is the only option to transplant it and get a new one to go into the body, or are there other options out there?
Another great question. As I said earlier, the ways we try to, treat cancer is traditionally three: you cut, you burn, you poison. You can cut it. You can cut out cancer if it's only growing in that little organ. But unfortunately, a lot of cancers spread. They spread to other parts of the body. So even if you cut it, if you cut the where it's growing from the little organ, it's hard to tell. It's hard to cut everything. You might as well kill the person, right? And so, in that case, you just poison. You give chemotherapy. If we can understand how to detect cancer super early when it's not spread, then the only option and the option that will cure most patients would just be to cut it out, to cut out the organs. Yeah.
And one last icing on the cake, what's the least dangerous type of cancer?
Well, I mean, there are many cancers now that have, very high cure rates. For example, testicular cancer has almost 100%. I would say 99%. Cure rate, and it doesn't spread as much. So it's one of the least dangerous kind of cancers. I'm not saying you should go out there and get it, but it's one of the least dangerous kind of cancers. Yeah.
Awesome. All right, Ian, I think you wanted to jump in and say something.
But so, and just to say, sorry, does someone have someone?
Someone has a question? Okay, no, I think not.
That for me was really fascinating to hear Toby talk about his work and cancer. And I think that, me listening to what he was saying there, I think there's several things that I just wanted to reflect on. Like, I really believe Toby is going to make really important contributions to understanding this disease and treating this disease. I'm really excited to see what he does in that way. But him talking about his own journey and goal stuff too. There's two things I wanted to highlight too. The first is that when I thought about science when I was a kid, you're sitting by yourself, in a dark room, or looking at Petri plates or something like that. Science is really interactive and communicative. And Toby started by talking about being at this institute, and he's actually literally just downstairs from us right now. And I love that process of just being able to talk and work with other people. And actually, science is very communicative, very collaborative. And so I started my own example of I love being able to work with Brittania. We get to talk about science and discuss how things are going and come up with plans and look at things together. And I really love having Toby as a colleague. And so he did such a great job of describing his science and the goal and this need to understand cells. And I think our lab's description would be pretty similar in a way.
Yeah.
Even though some of the experiments that we're doing are quite different in other ways. The other thing that he mentioned at the beginning that I just loved is what brings you into science and what gets you excited about it. And I think for many people that it's that desire to help people, to treat disease, to be able to combat those things. And I think that that's a really important component of what drives a lot of people. But I also really liked his statement of, what got him excited in the first place was just thinking about the cells and the cells in our body.
Yeah.
And I agree with that, right? And so we're going to see that today. Okay, so just thinking about what Toby said there, you know, he was talking about, you know, cells and cancer cells and your organs and tissues. You know, each of us, we all started as one cell, right? We all started with one cell, which is, you know, when sperm and an egg came together, that was where you began. Okay, just one cell. Our bodies now don't just have one cell, right? You have a lot of cells.
Yeah.
It's not thousands of cells. It's not millions of cells. It's not
billion.
billions of cells.
It's
It's trillions, trillions of cells, like 30 trillion cells. It's a number that I can't even comprehend, right? And so when you think about all of that and all those cancer cells behaving, it kind of, like Toby said, blows your mind. What we're going to go look at today is human cells growing in a dish. And so we're going to look at, you know, just a few cells at a time. We're going to look inside of those cells. Those cells are going to be alive. They're going to be like doing stuff and growing and moving. And I think for me, it was that time of actually being able to look down a microscope and see those cells that was like, wow, I need to study this, right?
Yeah, yeah.
And so we can't bring you all to our lab, but we're basically going to do the next best thing, right? So we're going to bring you all down the hall to our lab, and you know, Brittania and I will show you around a little bit for the lab, and then we'll look at down the microscope. And so we're going to share the microscope screen, and each of you, we can move around. You can tell us where to point, where to look, and we can see the cancer cells behave in a dish.
All right, so give us a few minutes. We'll meet you down the hall. Okay, I'm gonna try and walk with you, but the internet might go. Okay, guys.
You take that. I'll take the computer.
Okay.
Yeah, but you can. Yeah. Okay. So walking down the hall to.
All right. So we're heading down the hall. So we're gonna enter the lab space now. You see these big red doors. This is kind of where we get in and go inside. And our lab is all the way in the back. So we're gonna keep walking for a while. Are we still connected?
I don't know, but at least it's recording. Oh,
there you go.
Okay.
Okay. I think we just switched.
Okay. Do you want to show them briefly around our lab? And
yeah, sure. All right. So this is our lab space. We have one of our colleagues here. Hi, Asha. And you'll see it looks, you know, it seems a bit messy, but it's pretty organized in our minds.
What are you talking about? It's beautiful.
In our heads, it's pretty organized, right? And so here you'll see it runs all the way down this hallway, but there are different bays. So we have benches and stuff that runs in. And we also have more lab mates here. They kind of sit together. And you'll see that. So my lab bench is actually there. And it has lots of bottles and centrifuges that we use to spin, and flasks and beakers and all those wonderful things. So the bench right there is where most of the work happens. And then we have our desk where we do like computer work and all that wonderful stuff. There's a large refrigerator there where we keep all our reagents and all the stuff that we use to actually do our experiments. And it looks the same going down the hall almost. So we're gonna take you over to the tissue culture room. And this is where the cells get taken care of, right? So this is our TC room right now. And you'll see that there are incubators, there are different hoods that we have to actually keep the cells where everything is kept and all the media and wonderful stuff. So this is kind of the space that all the cells are. And this is where we take care of them, we change their media, and we ensure that everything is good to go. Is there anything else you wanted to show them?
You gotta show them the starfish.
Okay.
You gotta show them the starfish.
Okay.
So our lab mostly works with human cells and cancer cells, a lot like what Toby said, but we also have some starfish in the lab, which are amazing. And so here you can see the tanks of them, and they're growing in seawater and stuff like that. But
so they're right there. And yes, we do work on these, so we get to take them out and do experiments with them. And this is basically just a big cold room. So it's like a refrigerator that you walk into.
It's sort of like Boston in December, right?
Yes.
But all the time.
All right, now we're gonna head over to the microscope room. All right.
So it's dark because we're going to shine
Oh, well,
lights
it's
on there.
not.
And Kwon Chung is really nice. He set this up for us today. Okay, Kwon Chung, you want to mute your version so that we don't get the reverb? Okay,
right.
so you want to show the microscope and then we'll share the screen.
Yeah. So this is the microscope. Really big, really fancy. Looks very scary, but after working with it for a few times, you actually get the hang of, you know, what it does and how it works. I still don't know all the answers. And so sometimes I'm still like, can you help me to find this particular thing? But you get pretty used to it. So we are gonna share screen and we're gonna show you what that looks like for looking at live cells.
Okay.
You can admit session.
So sure, we can show it. Okay. So we're gonna look at this and let's see if we're gonna start putting here. Okay. So these are human cells, right? And so we're gonna look at these cells, and each circle shape there we see is actually a cell, right? And so we can move around in this, and in a second you guys can tell me where you want to move. And so each one of these shapes that we're seeing is a cell, but they have different fluorescent things, different things that we're shining light on where we can see stuff. And so these little dots we'll come back to in a second. I can focus up and down here on the microscope. Look up, you know, and so you can see like different parts of the focal plane. Hopefully you can see there's like in that cell, there's some different lines, okay? So now we're gonna, not that one. Oh, okay, and then got two. Okay, so this here, and again, I can focus up and down. This is the DNA in the cell. This is the genetic material, your DNA. You know, it's something that if you did a workshop with Brittania, you looked at on a gel and you thought about how you're gonna purify and isolate. This is the stuff that gives each of your cells instructions. And so what you can see is these round circles as we move around. Each one of these is what's called the nucleus, and it's where all of the DNA is sort of trapped and contained. You can see that it's not just, you know, smooth and even, but there's sort of little structures within that DNA as we move around, okay? And so then we saw in green, I think, that's the DNA, sorry. Ooh, okay. So now these dots right here, you can see, if you could see them, they're sort of overlapping. They're in that nucleus. They're on top of the DNA. These dots, that's what our lab works on. And so if we move around here, you can see that all these individual dots within the cells, these are a special structure in your DNA that's important for how cells divide. And so what's that? Different. Okay. And then this is that where we started, which I think is let's see if we can find a place that's not reached. There's like a lot of little dots here. These are part of the cell, and you can tell these are alive. So if we watch this for a second, let's see if we can find a place here that does a good job of showing that. Ooh, okay, this one's gonna be great. Look at this one here. Okay, so these cells are still growing in the dish. They're doing stuff as we watch them. And so if you pay attention to some of those dots in the cell, you can see that they're actually moving. And so there's actually like lots of things happening in your cells all the time. Lots of growth, lots of changes, lots of things moving around. And so this is a structure in the cell, happens to be called a microtubule. But you can see the tips of this. If you just choose one dot, hopefully you can see it sort of moving around and growing and changing. And let's find a cell that's divided. So maybe we'll go back to that one here. Okay. So this, ooh, yeah, let's check it out. Check it out. Okay. So this is a cell, that one, not the really bright one with the circle, but the sort of one, I'm going to move up the bright one out of the way. It's sort of in the right-hand side. This cell is a cell that's about to go through cell division. It's about to make two cells from just one that we're seeing. And so these dots there, these microtubules are providing the structure that does that. We can see up and down there. And let's watch the DNA here. See the DNA here, it's like dividing. And so it sort of compacted up, and you can see the cells, the DNA is sort of like separating itself from each other into two new cells. This is a process called mitosis, and these cells are almost at the end of that mitosis process here as well.
And I think they have a question for you, Toby. Is it the case that all the uncontrolled growth in cancerous cells are due to the activation of telomerase?
Can you say that again? Sorry.
Yeah. Is it the case that all uncontrolled growth in cancerous cells are due to activation of telomerase?
Well, it's not the case that it's due to telomere. I mean, increase in telomere length and increased expression of telomerase helps cancers grow, for basically, as long as the host lives. But there are other things that activate the growth of the cancer cells, such as what we call oncogenes. These are like the gas of the car. If you think of a cell as a car, it keeps the cells going. And then you have what you call tumor suppressors, which are, the brakes of a car, and they need to be broken for the cells to go crazy. So those two things particularly drive cancer, and telomerase helps to have the cancer grow even more.
Awesome. Thank you, Toby. And yes, the cells are real that we are looking at. They are alive. And
you can tell us where to go. Okay,
yeah.
so this one right here with this line of DNA, that's a cell that's just in the middle of its division process. So we can see all the DNA sort of aligned there. And those are those little dots, are nicely paired up dots. Those are, again, the structure that Brittania and I studied. And then this is what the microtubules look like in that. You see the two sort of bright things, we call those poles, and back and forth there. And so somebody now, you can control it. Somebody at home, choose where we should go. What do you want to look at? What do you want to see? They're live cells. And so you can control it like in real time to see what they look like.
Anybody has any suggestions? Do you want to go left, right? Where do you want to see? What do you want to see or look at?
Right.
Okay, right.
Oh,
Okay.
I forget, it's inverted when I did.
It's inverted, yeah.
I was pushing it right and it went left.
All right.
Okay, let's see. Any cell that you want to look at a little bit more closely and see what's going on with it?
I wonder if there's any cell that is, kind of in a process of mitosis.
White blood cells.
White blood cells. So this here is a plate of just a single cancer cell line. So it came from a cervical cancer from an amazing woman named Henrietta Lacks, who has more cells in the world than basically any single person these days.
Yeah.
But so there's not blood cells here, just Henrietta Lacks' cancer.
So there are no actual white blood cells present right now. I think something that's amazing, you'll notice here, right, how small the dots are. So if we go back to the kinetochore, which is kind of what our lab studies, you'll see that these are super, super tiny, but they're actually, a combination of multiple proteins or molecules like located at these small dots, right? So they're hundreds. Are there hundreds, Ian?
Hundreds.
Hundreds of different molecules that actually make up the kinetochore, and it helps with the cell division. So with the process of the cell going from one to two things, it really helps in controlling how that works and what that would look like.
So actually in this field right here, we can see lots of different stages of this. So hopefully you can see my cursor. This one right here, it's just beginning it. This one is just in the middle. We call this metaphase. And then this one up here is completing the process. It's separating out, called anaphase. And if we go back and look at those dots here as well, you can see all the dots. They're in the early one, the medium one, and the late one. But in this late one, they've sort of divided, and they're not these like paired dots anymore. Now they're sort of separated away from each other.
Anyone has any more questions or wants to know anything else about either cells or cancer or science in general?
Ooh, look how pretty that one is.
How does
Yes,
do
Ashley.
white blood cells like get to the like how do they like find where the like cut is the
Okay, so how do white blood cells actually locate where they should go in the body if we have a cut, for example, and a bacteria might have gone in? There are different chemicals that, or Toby might be better able to answer this, but there are actually different chemicals that are released, and it follows these different signals within the body, and that's how it actually locates where it should go or how it navigates around the body. Toby, you could share.
That is correct. Completely.
Okay.
Yeah.
Great question, Ashley.
How do you use the cells to know someone's blood type?
Okay. How do we use the cells to know someone's blood
Miss
type?
Moodie, before you diverge on that path, someone's just asking to clarify what the dots are called on the screen again.
Yeah, the dots are kinetochores. So that's what they're called. Does that make sense? They're called kinetochores, and it's a bunch of protein that is found that helps with cell division. All right. And so how do we use the cells to determine blood type? Well, that actually has to do with red blood cells and the proteins that are found on its surface. And you do a test with basically how blood clotting works, how the antigens interact to get the different blood types. So that's kind of how it would work in determining blood types, not particularly these cells that we're looking at right now.
Someone just asked one of my favorite questions, which is how do you get the cells to
Glow.
glow? And oh my goodness, it's so cool. Okay, so each of these things here, we're able to see inside these living cells because there are glowing fluorescent things in there. So if you imagine, if you ever had a glow stick and you break that and it like shines light, sort of what we're doing here is kind of the same. We're making fluorescent stuff that the microscopes can see. And so actually, if Brittania looks at her, let's change it to the GFP laser. If you look at on the microscope, and you can see on her camera, there's actually a blue laser sitting there, right? And so its job is to actually like turn on the cells. And so we're using a fluorescent protein. And so this is something that actually is going, and we're shining laser on there, and it's having special fluorescent light be emitted. So sort of like a glow stick that you might have, or a fluorescent sticker or something like that. But we're taking a protein actually from jellyfish. And so there's jellyfish that like glow and are fluorescent. And we took out that, not me, but scientists took out that protein, and now we can use it to sort of peer inside our cells by looking at this protein.
How are cells moving and they're not in the body?
So your cells are alive, and they need nutrients, they need oxygen, they need blood. But we can actually have cells grow in dishes in the lab. And so if Brittania shines that back there again, what we're seeing is not only the laser, but there's a plate there, there's a dish. And so the cells have food and they have other things to keep them happy. But when we're moving around like this, I'm actually moving the microscope and the stage. But we can watch the cells over time. They will move around and they'll grow and they'll divide and they'll do lots of things like that. And so it's possible to keep some cells, particularly cancer cells, which grow like weeds, unfortunately, happy in a dish going around and behaving too.
Can you control the rate they grow when outside the body? Yes, absolutely. And so, there are different conditions that you can control, and that's a part of science, right? We kind of determine what conditions do we want to follow or do we want to look at to figure out how that affects the growth of a particular cell, right? You could do things to make the growth slow down. You could do things to make it speed up. By adding different chemicals, of knocking out different proteins, and stuff like that. So yes, we can control. Can they die? Yes, they all do eventually die, in whether they're on the plates or whether they're in our bodies. There are cells that die all the time. Is the green fluorescent protein that you use? Yes. So green GFP, which is the green fluorescent protein, yes, that's what we use.
But we can use other colors
Yeah,
too.
we can use other colors.
So the way that we're seeing all these three different things in the same cells is that they each have a fluorescent color. And so we're using different lasers to turn them on at different times.
All right. Anyone have any? Yeah.
Can you make more cells out of other cells?
Yeah.
And,
Yes.
and that's also one of the biggest challenges of cancer, as Toby did such a nice job of describing at the beginning, is that these cells, they just keep dividing and growing. And so every time that we can see, one cell here, one of these circles of the nucleus, if we came back in one day, there would be 2 cells instead. And if we came back another day after that, there'd be 4 cells, and then 8, and then 16, and it just keeps growing, growing, and growing. And so this cancer is going to keep dividing. We can study that in the lab, but in a body, that's if a cancer cell is just going to keep growing in the way that it shouldn't, that's a big challenge.
How many cells grow a day? Wow.
So for most human cells, to go from 1 cell to 2 cells takes about 24 hours. But Toby was really nice. He gave us some of the pancreatic cancer cells that he uses. They take about 3 days to go from 1 cell to 2 cells. And so the rate can be quite different.
Can you shrink them? Can you shrink cells?
There are different cell sizes. And so these cells, like, okay, so if you imagine, let's say you're a meter tall. One of these cells is about 20 micrometers. And so it's 1 micrometer is 1,000 times smaller than more than 1,000.
1
That's
micron.
a million times smaller than a meter.
Yeah.
So these are much, much, much smaller than you, but cells can range in size from a range of different sizes, but you can't shrink them that much or they'll die.
Okay, I see. How are they looking at the cells right now? Is it some dried skin? It's not dried skin. So these cells were harvested a very, very long time ago and just passed down from lab to lab. And we keep them in like little vials or tubes. And we store them in very, very cold temperatures. All right. So it's like minus, I think 190 something. I don't know. But it depends, right? We store them in these freezers to keep them there. And when they're frozen, we take them out and we activate them by thawing them and adding media, so giving them food and stuff like that. Yes, so when we freeze them, they stop dividing, right?
But then when we're growing them in the lab, we can take them back out and we put them at the same temperature that your body is at.
Yes.
So you have your body temperature, hopefully you don't have a fever, you're about 37 degrees, and that's the same temperature we grow these cells at.
Can you give your cells to someone else? I mean, yes, I'm sure that's possible. That is possible.
One of my favorite things to do, if hopefully if you get to do a workshop with Brittania using microscopes, you can actually take your cheek cells, and it's super easy to look at your cheek cells. They're huge cells and they're so beautiful to be able to see. But yeah, you can look at your own cells someday too.
All right. Can they block blood from flowing through the body? So can cells block? Well, I mean, blood clotting takes place, and so red blood cells are actually red blood cells are actually there, and they can actually stop blood from flowing if they clot. All right, so that's one way that they could stop cells from running. Yes, the specific temperature is your body temperature that we keep them at, and that's 37 degrees Celsius. All right, guys, so we are gonna have to wrap up because I have a meeting to head to. We have lab meeting, and so we are gonna wrap things up. It was great having all of you guys. If you have any more questions, please feel free to let me know, and I'll ask Ian or Toby or anyone else in the lab to kind of give you a rundown on that. I'm glad that it was enjoyable. I'm glad that you had a good time and that you think this was a cool thing. We hope maybe one day you guys could actually visit me in person. I don't know how that would work, but sometime in the future probably.
And thanks also to Toby for taking all the time to tell us about
this was great. This is great. It was nice to meet you all.
and answering such fantastic answers and questions, and we really appreciate everything that Toby does. We're so lucky to have him as a colleague.
All right, everyone, have a good one. Bye.
Auntie British,
Yes.
I hope you have a wonderful day and I like it.
Oh, thank you, Jaden. All right, bye, everyone. If you would like more content like this, please subscribe to our YouTube channel, CYD Institute, and follow us on all social media platforms at CYD Institute, or visit our website, www.cydinstitute.com. Here, you could view our previous workshops and events and watch past videos. Head over to the gallery and you can view all our previous workshops and events. We hope that you'll join our community. Goodbye for now.